Welcome back to Medinare. In this video, let's discuss about Staphylococcus aureus. It is a gram-positive cocci, occasionally capsulated and spherical in nature and about 1 micrometers in diameter, and also it is arranged in grape-like clusters. This arrangement is because the cell division of this bacteria occurs in multiple planes with the daughter cells remaining close together. It is non-motile, non-sporing, facultative anaerobe, which means it can survive with or without oxygen. Also, this organism is catalase and coagulase test positive. Now let's discuss about the virulence factors. Those are the cell wall associated factors, toxins, and extracellular enzymes. The cell wall associated virulence factors of Staphylococcus aureus are peptidoglycan, teichoic acid, cell surface adhesins, protein A, and microcapsule. Let's look at each one of them. Peptidoglycan confers the rigidity to the cell wall and maintains the shape of it. It induces inflammatory response and also has endotoxin-like activity. Teichoic acid. It helps in adhesion of cocci to the mucosal surfaces and also it inhibits opsonization. Cell surface adhesins such as clumping factor or bound coagulase, fibronectin binding adhesin, and collagen binding adhesin are all virulent factors. Protein A also has many biological properties such as anti-complementary, chemotactic, mitogenic, inhibition of opsonization, and induction of platelet damage. It also mediates coagglutination reaction. Lastly, we have microcapsule. Some strains of Staphylococcus aureus have polysaccharide microcapsule which inhibits phagocytosis by neutrophils. The second type of virulent factors of these organisms are toxins. First toxin we are going to see is membrane active toxin. The Staphylococcus aureus produces 4 distinct types of hemolysins which are alpha, beta, gamma and delta. They are membrane damaging toxins, act on red blood cells leading to destruction of RBCs which is hemolysis. They differ from each other by their action on RBCs of different animals, their lethal and dermonecrotic and leukocidal activity. The next type of toxin is leukocidins, also known as pantin-valentin toxin or PV toxin. It has 2 components, namely F, which stands for fast, and S, which stands for slow component. Both the fragments act synergistically with the gamma hemolysin to damage leukocytes, RBCs, and macrophages. This gamma hemolysin and PV toxin are called as synergohymenotrophic toxins because they are not active individually, but in combination they are capable of producing hemolytic and leukocidal activity. The next type of toxin is epidermolytic toxin, also known as exfoliative toxin. This toxin is responsible for staphylococcal scaled skin syndrome. This syndrome may manifest with localized tender blisters and bullae formation. Also, Nikolsky sign is positive, which means exfoliation and separation of the outer epidermal layer, leaving the denuded underlying skin. The severe form of this syndrome in newborn is called Ritter syndrome. The next type of toxin is enterotoxin, which is responsible for staphylococcal food poisoning. Also, another toxin present is toxic shock syndrome toxin. As the name suggests, it is responsible for causing toxic shock syndrome. The extracellular enzymes which act as virulent factors are coagulase, heat-stable thermonuclease, deoxyribonuclease, staphylokinase, and hyaluronidase. Now let's look at the pathogenesis of Staphylococcus aureus. Initially, it gets colonized at various body surfaces such as anterior nares, axilla, and perineal skin. After getting colonized, they get introduced into the tissue as a result of any minor abrasions or any instrumentation which cause damage to the skin surface. Then they adhere to the tissue surfaces which is mediated by various adhesins, for example, clumping factors. Once they enter into the tissue, they get invaded into the tissue by elaborating enzymes like serine protease, hyaluronidases, thermonuclease, and lipases. These organisms then evade host's defense mechanisms by various immune evasion mechanisms such as antiphagocytic activity mediated by microcapsule and protein A, inhibition of leukocyte migration by chemotaxis inhibitory protein of Staphylococci, and intracellular survival inside the endothelial cells by formation of small colony variants. Once it evades the host's defense mechanism, it finally spreads to various distant sites by hematogenous spreads, resulting in various clinical manifestations. And that brings us to the end of the video. I hope you guys liked it. Do like this video and subscribe to Medinare for more. Thanks for watching.